Okay, so this first part of chapter 12 is basically just kind of going through like really basics of like what are tumors. So, here we talk about benign tumors. So they're like really well defined. Benign tumors are usually really well defined. Um, the only harm that they usually cause is like if they're pressing up against another organ and they're so large that they actually affect the function of another organ. So that's the big thing there. They're asking us to kind of understand like a benign tumor of the smooth muscle um, of the uterus is called a leioma. So, specifically, like a benign uterine tumor is called a leioma. Benign tumor of fat cells is called a lipoma. So, those are like probably two things to just try to like keep in mind. So, there's a picture of your leioma. So, here we're talking about malignant tumors. And like the key things about malignant tumors is definitely anaplasia, which is like a loss of cellular differentiation. So, this can occur like especially as cells stop looking like any other kind of cell and they sort of revert to this like almost stem cell morphology in a way. Malignant cells are also pleomorphic, which just means like their size and their shape can be all different. So these are some really good pictures of like cells going through anaplasia or pleomorphism. Like we have different sizes, we have different shapes. We also just have like, what are these cells? The problem with malignant tumors is like, if you're able to find the sentinel tumor, which is like where it originates, for instance, like, like a lymphoma, it's going to start in lymph tissue as it kind of like morphs and like metastasizes, it's sort of just like doesn't really look like anything. Um, so the big one they're highlighting here is carcinomas. So cancers arising in epithelial tissues. So that's like, your skin tissues or your pleural linings, your membranes, and a big one they want us to remember is mammary adenocarcinoma. So that's a malignant tumor that comes from, let me highlight this, breast glandular tissue. So basically, you have like, um, oh my gosh, what are they? Ugh. Glands in your in your mammary glands. So mammary adenocarcinoma. Sarcoma is like, it's from the mesenchymal tissue. So I don't know if you remember when we were studying like fetuses and how they grow, and they have like the mesenchymal, they have, which is like the middle layer, which turns into like muscle and connective tissue. And then there's like the two other layers that are like the internal layer and the external layer. So sarcomas come from that middle layer, which becomes like your muscle and bone, connective tissue. Lymphomas are lymphatic tissue, so that's going to be cancer of lymphatic, and then cancers of blood forming cells are leukemias. So that's pretty much those first couple paragraphs. Carcinoma in situ, it's like, this is kind of a weird one. I sort of struggle with this one. Refers to pre-invasive epithelial tumors of glandular or squamous cell origin. So they're basically saying like, think of like skin, like you usually get like in skin, like really early stages of skin cancer, especially in like the more mature population, like 50s or 60s. Like they didn't wear a lot of sunscreen when they were children, so they get a lot of those like squamous cell like mole things, and they just basically just like get them removed. But that's what they're talking about. It's like a carcinoma in situ is basically like, it is an epithelial tissue tumor. It's malignant, but it just hasn't grown like outside of its little area. Their early stage cancers are localized to epithelium. So exactly like those little skin cancer moles, that would be an example of like a carcinoma in situ. So that's basically all of that. And then they're just talking about like, you can have high grade, low grade dysplasia. So like if you've ever, like when you have what is it? HPV, and the biggest strain, the biggest genetic strain of that one is strain number 16, which leads to like uterine or like penile cancers. And so that would be like if you had HPV 16 and then you had like a low grade or a high grade dysplasia, that could be a carcinoma in situ situation. So you got to get that removed. Biology, let's see if there's anything here. And blah, blah, blah. The analysis remains a leading overview of why cells malignant. The following discussion is organized. Oh, these are hallmarks of cancer. So like basically, I feel like what's important about this is just like, if you're trying to grade something that's like malignant, you're going to be looking for like, it's got genomic instability, like it's got one of those mutations like a BRCA1 or a Philadelphia translocation. So remember everything we kind of talked about like in genetics chapter. So this is where we're going to start to layer genetics into actual disease states that make sense. So we can have genomic instability like translocation, deletion, a mutation, inducing angiogenesis. So that means like the tumor is creating its own like blood vessels, its own supply, resisting cellular death. So basically it can resist apoptosis, deregulating cellular energetics. I'm not quite sure what that's referring to. Sustaining proliferative signaling. So this is a really big one. These malignant tumors can basically just like they have autocrine signaling. So they basically, this is actually really cool, and we'll get into this, is like they can signal themselves growth factor hormones to literally just keep growing, but they can also start to basically signal to their like the neighboring cells around them to start proliferating. Like they can literally override the homeostatic mechanisms of like other normal cells around them to basically start proliferating. Like it's really crazy. So basically the rest of this chapter is going to be going through all these like details of like hallmarks of cancer basically. So now we're talking about as far as like genetic stuff, like point mutations. So this is basically like, you know, those nucleotides, which basically make up like the rungs of the ladder of like DNA, RNA. So that's like if you had one of the nucleotides, like basically just get like change with another one. So they like kind of, let's see, mutations include point mutation, small scale changes in DNA, alteration of one or more nucleotide base pairs. So it's literally just like a protein gets one of the wrong base pairs. This type of profound effects on result in proteins. Yeah, so like a point mutation is like, let's say you're making a protein nucleotide codon, which is like 3 of the nucleotides, and you're coding for a heart cell, you know, and let's say one of those 3 nucleotides in that codon, a codon is a set of 3 nucleotides that codes for a specific amino acid. Let's say one of those nucleotides gets like it's the wrong one, like it's not guanine, it becomes adenine. So you basically have the incorrect nucleotide. So now that amino acid is going to potentially have malignant consequences in like the heart, for instance. Chromosomal translocations. So this is like when in the Philadelphia translocation, when chromosome 9 and chromosome 22 basically switch places. And so that can lead to different types of cancer. Gene amplification is like when there's like one specific part of a chromosome that just keeps getting replicated over and over and over again. So that's like an inappropriate replication, making multiple copies of this one gene, you know, and that's not good either. So DNA methylation, that's basically, remember the on-off switch for genes. Like when something gets hypermethylated, it's like, I think it's turned on. And the histone acetylation, that's the same thing. It affects that off-on switch. Driver mutations, they're, let's see, what are they? Small nose, individual cancer. Not all mutations malignant. Some called passenger mutations are just along for a ride. Okay, so what I'm understanding is like basically whatever version of mutation happens, if it starts to develop into like a malignancy of some kind, or it starts to move into clonal proliferation or clonal expansion, like at a malignant pace, that's what makes it like a driver mutation. Okay, let's keep going. So we have malignant transformation where normal cells become a cancer cell. So the reason why they kind of brought up like chapter 11 before this chapter is because they were trying to show that like stress, like chronic stress, like let's say cigarette smoke on your esophagus can lead to Barrett's esophagus, which can lead to lung cancer. So they're really talking about malignant transformation occurs through like basically environmental stress that you're putting on your own body that can turn malignant. Like cancer is a weird one because there is the genetic component, but it's really heavily influenced by like how you're treating your body. So that's why they kind of talk about stress before all of this, because, stress includes UV radiation from not wearing sunscreen. It includes smoking. It includes, maybe like not eating the correct foods. So anything that you're causing additional stress on your body to the point that cells are adapting to that climate will potentially turn it into like a cancerous situation. So that's kind of like what they're talking about. And of course, like you're going to have like inflammation and stuff. And that's going to be primarily mediated by T lymphocytes and macrophages, B cells and neutrophils. So basically what happens is like you are putting your body under so much stress. And remember, like when it's releasing long-term cortisol, you start to get like inflammation, like we kind of saw in like the Cushing syndrome. So this creates stroma, which is a tumor microenvironment. Like it basically helps it to become cancerous, and it helps with proliferation. I'm not worrying about this because I haven't seen it literally in any quiz question. So I'm not worrying about that right now. Genomic hallmarks. All of this stuff, this is really dense stuff. I did not really see it. I saw Ross. So Ross is definitely an important one. That one came up. It's an acronym for rat sarcoma, was found originally. EGF receptor activation linkage by bridging protein activates Ross via the replacement of guanine diphosphate with guanine triphosphate. Activated Ross then initiates signaling. Okay. Operation. Okay. Oh, okay. So here's what they're basically saying. So all of these things are pathways inside of the cell. So they all do like different things. They phosphorylate cytoplasmic proteins. So basically they add on like phosphate groups to proteins, which the phosphate groups, when you add them to proteins, is kind of like they add to like the identity of the protein kind of. So basically what happens, Ross comes in, this rat sarcoma, think of like a little rat. Like, oh, he's the rat of the group. He's going to go to the cops and he's going to, like literally like a little rat comes into the cell, starts activating all of these things that basically lead to the cell inappropriately proliferating and growing. So proliferation can be discontinued through spasm by a decreased level of growth factor. Okay, so they're basically saying RAS is something that gets all of these pathways to start proliferating and growing. And there are stop gates that can stop it. But guess what? Probably if they can't be stopped, then they're going to turn into cancer, right? Genes that encode pathways design called proto-oncogenes. So proto-oncogenes are like they're pro-growth. Cancerous cells characteristically express mutated or overexpressed proto-oncogenes, which are oncogenes. So we kind of have this like, these are proto-oncogenes. They turn into oncogenes. They're pro-cancerous, essentially. Like, that's how I think of it. Let's see. This is what we're just talking about, autocrine stimulation. So basically, cancer cells have the ability to stimulate themselves. So that's like the self-autocrine stimulation. So they can stimulate themselves to grow. Breast cancer production of the human epidermal growth factor receptor, also known as, is upregulated as hyperresponsive low levels of EGF. Okay. Receptor turn. Oh my god. MYC proto-oncogene found on chromosome 8 is normally activated. This one came up like once. So I'm gonna highlight that. Basically, what we're talking about, this is a really dense section, but all it's really talking about is just all these different pathways that lead essentially to cellular proliferation and differentiation. And these things are basically called proto-oncogenes. So they benefit cancer because they basically proliferate growth. They have different ways that they could be stopped, like a ROS2 inhibitor, I think. And essentially, like, if they don't get stopped, they just become oncogenes, which are basically like, we're going to become cancer. So here we're now talking about like Philadelphia chromosome. So the translocation fuses two chromosomes in the middle of two different genes, BCR on chromosome 9 and ABL on chromosome 22. So basically chromosome 9 and chromosome 22 fuse together and they create a BCR-ABL fusion protein. So this basically just promotes inappropriate growth of like, myeloid stands for like rat. So, but I think it's like muscle cells. So that's bad. So basically the big thing here, Philadelphia chromosome is a translocation of chromosome 9 and 22, which literally creates this BCR-ABL fusion protein, which basically turns into cancer. Promotes the growth of myeloid cells. Evading growth suppressors. So we have tumor suppressor genes which regulate the cell cycle. They are called anti-oncogenes, which makes sense. They're not proto-oncogenes, they're anti. But these tumor suppressor genes can obviously get screwed up. Comparison of cancer genes has the caretaker, dominant oncogenes. I don't know what this table is about. I'm ignoring it. All right, so the big tumor suppressor gene that we've learned about, especially like in chapters 4, 5, and 6 were about genetics, was the TP53. So the TP53 is like a really, really big one. This is definitely one to at least just be able to recognize and be like, yes, this one is a tumor suppressor gene, and when it basically gets overridden, cancer happens. So this is in the nucleus of cells in the body and attaches directly to DNA. When DNA becomes damaged, the protein plays a critical role in whether the DNA will be repaired or damaged cell will die from apoptosis. So basically what these things do, these tumor suppressor genes, they're like, hey, we're attached to the DNA, we're making sure everything's cool, and depending like if the DNA gets damaged by chemical or oxidation or reactive oxygen species or just DNA damage that can happen from cellular injury, this P53, TP53, is basically like the decision maker. They're like, okay, is this DNA salvageable, or is it to the point where we just need to trash this whole cell and have it die? So P53, like we just kind of talked about, detects anything that causes cellular stress, anoxia, oncogene expression, nuclear damage, and cellular stress is communicated via intracellular signals. Let's keep going. So these are basically just like all ways that like cellular senescence is basically when it gets to the point where it's running out of telomeres, which are like, just imagine them like the plastic piece on your shoelace. Basically just prevents, like, as you get older, your telomeres shorten, so you have that many fewer times that your cells can divide, which is part of old age. Like, that's how old age happens. Um, let's see, what does that say? Oh, this is an important one. This one came up in the adaptive quizzes. A very rare inheritable loss of function mutation in TP53, resulting in a 25-fold increase of developing malignancy. Okay, so basically in Li-Fraumeni syndrome, the big thing to recognize is to associate this weird word with TP53. And basically what happens is it like just screws it up. So it can develop into all these different types of cancer. So if you can just recognize, oh, Li-Fraumeni, TP53, you can answer a question. That's kind of literally how I study, is I'm just like, okay, what are they associating like germ cell mutation with, whatever. Like, I just try to make quick associations. Epigenetic silencing, modulating of gene function. They also have association modification. Okay, so this is where I was getting confused earlier with DNA methylation and the histone stuff. So that leads to the silencing of a gene. So you could like have the silencing of TP53 through like DNA methylation. Please don't ask me to define methylation right now. It's basically just like when on a chromosome, phosphate just keeps getting added on to like a specific gene. So it just gets like overly phosphorylated, which basically just like turns it off. So this is actually, that's also part of like cell differentiation, how we get like a muscle cell versus, another type of cell. Retinoblastoma is on chromosome 13. Remember that. That was on a lot of the adaptive quizzes. This is seen a lot in like children, though. That's what I remember about that. It's anti-growth and blocking activation of growth in the cell cycle. Mutation can lead to persistent cell growth. Have a subtle mutation, such as a point mutation. Inactivated retinoblastoma. So basically, just remember retinoblastoma is chromosome 13, and how it can kind of have like really subtle mutations is like through a point mutation. But remember that it's a tumor suppressor gene. So we don't really want that one to get screwed up. And it's related to the eyes. Children with retinoblastoma have the inheritable form, and most will develop tumors in both eyes and are at increased risk for secondary primary cancers such as sarcoma or malignant melanoma. So that's what I remember reading about. Retinoblastoma is like, it's kind of really related to like children. Wilms tumor, remember this one. This one came up a lot. And specifically that it's a childhood cancer of the kidney WTI gene. This one came up a lot. The Wilms tumor is part of it. It creates basically a tumor in the kidney. Neurofibromatosis, NF1 gene, and this all came up in the quizzes. So they care about Wilms tumor being associated with kidney tumors in childhood cancer. They care about neurofibromatosis being an NF1 gene. And the APC gene is part of like the polyposis colorectal cancer. So those all were really like, they kept getting repeated over and over and over again. Genomic instability, pretty simple. It's like all the genetic alterations that we've talked about. MicroRNAs are basically like regulator RNAs. So they regulate the gene expression by targeting messenger RNA. So they can basically like repress DNA translation, which is good because if DNA is screwed up, it's a problem. Oncomeres promote multiple cancer pathways. So oncomeres are bad. Oncomeres are bad. Let's see. Let me just like quickly run through and see if there was anything else. Oh yeah, here's telomeres are protective ends or caps of repeating hexanucleotides on each chromosome and are placed and maintained by a specialized enzyme called telomerase. So like I said, they're like kind of like, just imagine like the shoe caps on the end of shoelaces. So short telomeres usually signal the cell to cease cell division. So your telomeres become shorter and shorter and shorter, which basically is how we start aging. They're mostly found in stem cells and germ cells. Cancer stem cells is a subpopulation of tumor cells that have replication and mortality because, oh, I remember this part. Okay, so basically, oh my gosh, all right. This video is already 26 minutes, so I'm going to upload this and then I'm going to do a second video that basically just finishes the chapter.